There have been enough changes to our understanding of coronavirus replication that we need to re-record this part. So that's what you're watching right now. Coronaviruses replicate in and around double membrane vesicles. And so that's these things that are highlighted in red. They have two lipid bilayers around them. And as we'll see, they're actually like a collection of flattened out vesicles that are stapled together at these points that are called the DMV pores. It's not that the pores are punching through anything, it's that they're joining up multiple smaller units to make a paired membrane, double membrane vesicle like this. Nearby, you'll see what are probably the makings of these. It's paired membranes, and most likely these are paired because they have pores in them, same way. It's just that these haven't been joined up into double membrane vesicles the way the red ones have. So these are called convoluted membranes, and over here these are budding virions. Alright, so the new understanding comes from a beautiful structure, and that's the thing we have rocking above us right here. This is the structure of the DMV pore, or at least as much as we have of it right now. This is made of 12 copies of NSP3, 12 copies of NSP4, and in the real thing, there are probably also 12 copies of NSP6 in there. They were not solved in this structure, but we have some reasons to believe that it should be there as well. So we won't talk more about it because we don't know exactly what role it plays. Interpreting this diagram, though, it looks as though it should be on the outside of that ring. That's the most likely position for it. All right. So the DMV pore looks like a crown, and it's got 6 little prongs on it, and each of the 6 prongs is made up of 2 NSP3s. NSP4 is mostly going to be sitting in the membrane. You can't see the membrane here, but there's a membrane here and a membrane here, and those are what are being joined together. Now what's actually going on is more like this, and so if you look over here on the side, so here's what NSP3 is doing. It has, we used to think it had 2 transmembranes. It actually has 4 transmembranes in NSP3. And so those are going to go through. There's an ectodomain, which means the part that's going to stick out into the ER lumen. And then there's NSP4, which is going to have an ectodomain here. And the idea is that the ectodomains of NSP3 and NSP4 may be what's coming together. Or it may be that 2 ectodomains of NSP4 are what's coming together. I think that's more likely based on some experiments that we've done. And what that's going to do is that is going to join up 2 sides of the same thing. So these are both interacting on the same face. It's like they were translated here as one big protein, polyprotein 1A, or the first part of polyprotein 1AB. They're cleaved, and the cleavage between NSP3 and NSP4 is done by the papain-like protease of NSP3 in SARS-CoV-2. And then if these 2 things interact with each other, they're going to create a pinch in the membrane. They're going to staple 2 things that are next to each other into a sort of an awkward sort of pinch shape like that. If these things are happening and you get basically 12 of these interactions, or 6 interactions between pairs of NSP2 and 4, then together those will form the 6-sided pore that you see here. And all they're doing is they're just sitting at the edge of vesicles, and they're all banding together. So a double membrane vesicle isn't one vesicle, it's a whole bunch of little vesicles that are stapled together at the edges like that. From the other experiments with infectious bronchitis virus, we know that NSP4 is enough to pair up membranes in the same way as they normally pair up. Oh, the other interesting feature is that NSP4 has 1 transmembrane that has this long ectodomain, and it has 5 other transmembranes. And that first transmembrane is actually going to go, and it's going to end up all the way over here, we think. So that's going to end up in what is basically the outside of the pore, and the rest of it's going to end up making up what is more or less the inside of the pore. There's a domain of NSP3 right near the end here, this C-terminal domain. That thing is going to end up forming, it's like a plug in the center of the pore, and so that is going to control, and we don't know what control means in this case, but it's in position where it would regulate what goes in and what comes out of the pore. In addition, there are many domains all up and down this sort of green part here that I'm circling that have RNA binding affinity, like good affinity for RNA, or have zinc fingers and stuff like that. And so most likely, however the RNA gets out, it may be the helicase is pumping it out, it may be that NSP3 Y domain is doing something to bring it out. Once it's out, it's got sort of a sticky trap that'll catch it and protect it. And then it's also been observed that there are swarms of nucleoproteins that hang out in the vicinity of the nuclear pore. And so these are likely to be there to catch the RNA. How do they get there? What are they being held by? There are still a lot of open questions here, but that's our current understanding of the pore. So let's go look at structures of the pore. Here we go. This is from the cryo-EM structure. And what you see here and here are like a fitted model where you try and put the protein inside the density you get from cryo-EM. So yeah, you can see the general structure of it and the way the proteins are hooked together. And there are gaps here. You may be just able to see them. There's a little dotted line here. That's a part that is actually connected in the protein, but it is too disordered for them to get a structure of what's happening in there. So it may be just a flexible bit between those two parts. Hard to say. All right, there we go. Okay, and let's look at a little bit of the bioinformatics of NSP3 and NSP4, because that helps understand what's going on here. So when I say Y domain, it's these things back here. These are the C-terminal domain. There's a transmembrane region either side of the ectodomain, so it's just transmembrane 1 region and 2. This one has 3 little transmembranes very close together. This one has 1 transmembrane, and these are the things that are going to hold NSP3 in the membrane. The NSP3 ectodomain, I think, most likely pairs with other NSP3 ectodomains, but it may pair with the NSP4 ectodomain as well. Everything you see up here, this is going to form the prongs, the big things that stick up, like this big sort of green things that you can see here. The orange ones are only sticking up half as far just because the rest of that protein is unresolved. It wasn't consistent enough in its positioning for them to be able to solve it. And so what you've got here are a bunch of little domains, like NAB is a high affinity nucleic acid binding domain that'll actually separate double-stranded RNA into single-stranded RNA. So it's a thing called an RNA chaperone. The papain-like protease 1 and 2, these things have a zinc finger in them, which would stick to RNA, and is not relevant for the papain-like protease function. When a virus has 2 papain-like proteases, usually one of them, PL2-Pro, is going to cut here, and I believe PL1-Pro is going to cut upstream of NSP3. Let's see. There are 2 ubiquitin-like domains, and both these papain-like proteases are actually pretty good deubiquitinases, and so we don't understand completely. There have been some experiments where people mess with the ubiquitin domains, and it does have impacts on virus growth and on the intracellular immune response. I don't think there's a good consensus yet on exactly what they're doing, but these are very nice little structural mimics of ubiquitins. So decoys of some type is all I can imagine, or things that would maybe bind to ubiquitin ligases that the virus wants to target for destruction. It's another possibility. DPUP is just a name for a domain. We don't know what it does yet. But these 3, MAC1, 2, and 3, are, well, one of them at least, is an ADP ribose 1 double prime phosphatase. and that is an enzyme that is able to, all of these are able to bind to RNA very nicely. This one will also bind to ADP ribose and will kind of slowly, kind of inefficiently cut ADP ribose, poly ADP ribose rather, into mono ADP ribose. Yeah. HVR is a hypervariable region, and it's just a thing that is there. Experiments that have knocked it out and messed with it show it doesn't really do anything. The main function would be that it would lengthen the length of these little prongs, and so it may just be a spacer there. And you see a lot of variability even among members of the same species of coronavirus. So those are the parts. And then the Y domain here is your plug that's going to be on the inside, right down here by the membrane. All right. What about the rest of the things? So, main protease. So that was 3. This is 4, 5, 6. Main protease sits in between these things. Main protease cuts itself off from these things, but there's no good reason why it would have to be there. It's just a thing that is always in this position between the NSP4 equivalent and the NSP6 equivalent. That's just where you find a main protease in a coronavirus or their relatives, all the way down to the invertebrate nidoviruses. And these are things that are conserved. There's always an NSP4 equivalent, like a bunch of transmembranes right before NSP5, and there's always a bunch of transmembranes after NSP5 that are probably the NSP6. And there's usually a pile of transmembranes that come before the NSP4 equivalent, which would be the NSP3 equivalent. And you find those in everything that is a nidovirus. And so it would appear that the DMV pore, like the things that make the DMV pore, seem to be conserved. So we don't know yet for sure, but it is most likely that there is something like a DMV pore in everything that is a nidovirus. So it's fundamental to how they work. All right. Here is the transmembrane, and our understanding was that there were 3 transmembranes here, and now we know there are 5. Yeah, we were undercounting. This is transmembrane 1 that's actually going to end up on the other side of the membrane over by NSP3. And these 3 are going to end up in the bottom membrane mostly, so they're going to be pointing into the DMV. In between, we have the NSP4 ectodomain, which is important for joining together membranes, even all by itself, even if there's no NSP3 there. 4 will interact with 3 if they are there. That's how they formed this lovely pore, which was made by expressing 3 and 4. 6 may interact with NSP4, but it has been very difficult to show that that's actually the case. It's still just a thing that's suspected more than known. But as you can see here from the little colored bands, there aren't many big gaps here. And you get nice colored bands that run top to bottom. And all that's saying is that generally everything's conserved in this area. These are important enough parts of the coronavirus replication cycle that generally there's not much evolutionary tampering with them that is tolerated. And the main protease sits in between. It's got 3 domains. These first 2 are the active site. They form 2 beta barrels, and they're hooked up kind of sideways to each other, like one sideways and the other would be open and looking at you. Yeah, there we go. Yeah, and the active site's right where those 2 barrels come together, and that's a chymotrypsin enzyme shape. So we call this a chymotrypsin-like enzyme, or you can call it an enzyme like the 3C protease of picornaviruses, which was real famous back in the day, but it's actually specific to poliovirus, this particular name for it. And now I think coronavirus is far more present in the consciousness. So maybe they should call, the 3C protease the main protease-like protease of picornaviruses. I don't know. Domain 3 is a dimerization domain, so it's going to link 2 of these NSP5s together. And that's fine. That helps NSP5 do its job. That's enough on the components. NSP4 will form so that you're going to get pairs of them. And so here's a pair, this pink one and this sort of more fuchsia one. Maybe lavender and fuchsia, I don't know. Yeah, Colors are hard. So this is another pair. This is a third pair. And so you can see it's not making a hexamer. It's actually 3 pairs that are along the inside, and then the pore would be dead center of that.